In aerobic conditions, pyruvate is oxidized to carbon dioxide and water, and NADH is reoxidized to NAD plus by the passage of its electrons to oxygen in mitochondria. But in anaerobic conditions, like in very active skeletal muscles, in submerged plant tissues, in lactic acid bacteria, NADH generated by glycolysis cannot be reoxidized to oxygen. If NAD plus is not regenerated, then the cell is left with no electron acceptor for the oxidation of glyceraldehyde 3 phosphate, and the energy yielding reactions of glycolysis stop. Therefore, NAD plus must be regenerated. Fermentation is the process that uses organic molecule such as lactate or ethanol as the final electron acceptor to regenerate NAD plus from NADH so that glycolysis can continue. First, the lactic acid fermentation. In animal tissues, in absence of sufficient oxygen, NAD plus is regenerated from NADH by the reduction of pyruvate to lactate. Also, in some tissues and cell types, such as erythrocytes, which have no mitochondria and thus cannot oxidize pyruvate to carbon dioxide, produce lactate from glucose even under aerobic conditions. This reduction of pyruvate to lactate is catalyzed by lactate dehydrogenase enzyme. We know that in glycolysis, dehydrogenation of two molecules of glyceraldehyde 3 phosphate derived from each molecule of glucose converts two molecules of NAD plus to 2 of NADH. Because the reduction of two molecules of pyruvate to 2 of lactate also regenerates two molecules of NAD plus, there is no net change in NAD plus or NADH. The lactate formed by the active skeletal muscles or by erythrocytes can be recycled. It is carried in the blood to the liver where it is converted to glucose during the recovery from strenuous muscular activity. This is known as Cory's cycle. Although conversion of glucose to lactate includes two oxidation reduction steps, there is no net change in the oxidation state of carbon. Both in the reactant and product, that is in the glucose and lactic acid, the H is to C ratio is the same. However, some of the energy of glucose molecule is extracted in the process, which is enough to give a net yield of 2 molecules of ATP for every glucose molecule consumed. Fermentation is thus a process which extracts energy as ATP, do not consume oxygen, or change the concentration of NAD plus or NADH. Alcoholic fermentation. Yeast and other microorganisms ferment glucose to ethanol and carbon dioxide rather than to lactate. Pyruvate is converted to ethanol and carbon dioxide in two-step process. In the first step, pyruvate is decarboxylated in an irreversible reaction, and this is catalyzed by enzyme pyruvate decarboxylase. This enzyme requires magnesium ion and has a tightly bound coenzyme thymine pyrophosphate to perform its activity. In the second step, acetaldehyde is reduced to ethanol through the action of alcohol dehydrogenase enzyme. With the reducing power furnished by NADH derived from dehydrogenation of glyceraldehyde 3 phosphate. As in lactic acid fermentation, in alcoholic fermentation also, there is no net change in the ratio of hydrogen to carbon atoms when glucose is fermented to 2 ethanol and 2 carbon dioxide molecules. Therefore, in all fermentation reactions, the H is to C ratio of the reactants and products remains the same. The enzyme pyruvate decarboxylase is present in brewers and bakers' yeast and in all other organisms that ferment glucose to ethanol, including some plants. The carbon dioxide produced by pyruvate decarboxylation in brewer's yeast is responsible for the characteristic carbonation of champagne. In baking also, carbon dioxide released by pyruvate decarboxylase when yeast is mixed with the fermentable sugar causes the dough to rise. This enzyme is absent in vertebrate tissues and in other organisms that carry out lactic acid fermentation.